Well, welcome back to Bio 180. Today, for our lesson, we're going to be having as our primary topic the Calvin cycle. This is going to be the second half of photosynthesis. And so I'd like to start out, we're going to review the light reactions that we learned about last time quickly, and then move on and see how they connect with the Calvin cycle. And we'll introduce and explain the Calvin cycle itself. And then we'll end up talking about two different problems that arise in the Calvin cycle. One of them related to stoichiometry, with the solution being cyclic electron flow. And then the final thing that we'll talk about will be photorespiration. So let's get started by coming over here and looking at the light reactions. And so I've drawn up just a representation of the light reactions. You may recognize some of the different proteins and players that we have up here on the board. Over here on this side is our photosystem 2 complex. This large rectangle is cytochrome BF complex. This is our proton pump. And then photosystem 1 down here at the end. We've got another enzyme, membrane-bound enzyme, NADP plus reductase. This is where we're going to make our NADPH. And on the very end, we've got ATP synthase. Ferredoxin, plastocyanin, and plastoquinone are all carriers or shuttles that will transfer electrons between these major complexes. And then over here, we have sort of our starting point with the water splitting complex, and this is going to be the source of our electrons as we split water. And so our process is going to start at photosystem 2 at the region called P680. That's the reaction center. And we're going to have a photon of light that comes in, excites P680 to P680 star, which then donates its electron up through the photosystem 2, and eventually that electron ends up on plastoquinone. Now P680, we just said it went to P680 star, its excited state. When it loses that electron, it comes down to P680 plus with a positive charge. And P680 plus has to grab some electrons in order to finish its cycle. So it's going to grab electrons from the water splitting complex. We're going to take a water molecule, and as the name implies, we're going to split it. We're actually going to take two water molecules. And we're going to take the two oxygens and we're going to form an O2 out of it. Out of that, we get 4 electrons. And those 4 electrons are going to be what we feed one at a time into the P680 cycle and send those electrons through our electron transport chain. And then we're also going to get, each water has 2 hydrogens, and we've got 2 of them. So we actually get 4 protons or hydrogen ions over here too. We'll come back to these in a moment. So that's where we're replenishing our electrons from. P680 again grabs them, donates them to plastoquinone. Plastoquinone donates them to the cytochrome BF complex. And they sort of move around the cytochrome BF complex and get passed through several carriers. In the process of that happening, we are going to take some protons that are here in this region is the stroma. So there's protons located in the stroma, and we're going to pump them into the thylakoid space. And so we're going to get some hydrogen ions or protons that get pumped into the thylakoid space. As the electrons continue their movement, they're going to be passed on to plastocyanin, which just simply acts as a shuttle, to pass them to photosystem 1. Photosystem 1 has its reaction center called P700. P700, as a photon of light comes in, will excite the P700 reaction center to form P700 star with a little asterisk there on the top. P700 star takes that excited electron and donates it to ferredoxin. And then in order to replenish P700, so we get P700 plus, this electron from plastocyanin is going to come down and replenish that P700 plus and allow this cycle to happen all over again. In the meantime, the electron that was excited and shuttled off goes to ferredoxin. Ferredoxin passes it to the NADP plus reductase, and we have, as its name implies, NADP plus. This cofactor comes in. It actually picks up those electrons and forms NADPH. And that is one of our primary products that comes off of the cyclic comes off of this electron flow. One small note, it actually, though we've been showing just one electron being passed, right here it actually requires 2 electrons in order to make one NADH. So you have to get 2 electrons that get passed from it. Remember, we started over here with 4 electrons. So as we're passing them through, 2 will collect and make 1 NADPH, and then the other 2 out of those 4 will come through and be used to make a second NADPH. So that's a little bit about the amount of NADPH we make. So all of this electron movement through here has done 2 things for us. One is we have pumped protons. We've used that electron flow, like electricity, to power this proton pump and move protons against their gradient into the thylakoid space. And it's about every 1 electron that goes through gets 1 proton pumped through. So that's the first thing we've done, is pump protons. And the other thing we've done is we've created this NADPH, okay, which actually is a high energy molecule. We took the sunlight. The way we were able to do that was from the energy of sun powering these electrons, kicking them up, and allowing them to land on NADPH. However, there's one other thing we're going to do. We're going to take these protons that were created from the splitting of water, and we're going to take these protons that were created from the pump, and we are going to run both of those, or all of those, both of those groups, through ATP synthase. So they'll come and run through the carrier, and then we're going to take ADP plus inorganic phosphate, and as that carousel runs, as we've talked about it before, we'll get ATP. And that's going to be our other product. We have enough protons here, 8, and that provides us enough to make actually 2 ATP. So as we run through this cycle, for every 2 waters, we're going to end up with 2 NADPHs and 2 ATPs. And that will become important later as we talk about the Calvin cycle itself and the ATP and the NADPH that it uses. And that's going to be the final spot for us. This NADPH and this ATP are going to remain in the chloroplast, and they'll be shuttled into the Calvin cycle. So now that we know where the NADPH and the ATP come from, let's talk about the Calvin cycle itself. So the primary purpose of the Calvin cycle, this is where we are going to use, so we're going to consume ATP and NADPH that was produced over in the light reactions. We're also going to consume CO2. So this is how we're going to take thin air, the CO2 in the air, and we are going to incorporate it to start making organic or biological molecules. So we're going to consume our CO2 right here, and we are going to produce, a lot of people, a lot of books, you'll just read it, we're going to produce sugar. That's not totally true. We actually are producing part of a sugar. We are going to produce a molecule called glyceraldehyde, 3-phosphate. And that glyceraldehyde 3-phosphate is going to be transported into the cytosol and used to make glucose in the end. So we're actually producing about half a sugar in this. But it does allow that plant to make glucose, which is the ultimate end goal. So that's what we're going to do with the Calvin cycle. So the Calvin cycle is going to start out with a molecule called ribulose bisphosphate. And some important things to note about it. As it implies, it's been energized by the addition of 2 phosphates on it. Ribulose is a 5-carbon sugar, and that's also significant. And so this is going to be the starting point of our Calvin cycle. And in the first phase, we're going to take our 5-carbon ribulose bisphosphate, and we are going to add a molecule of CO2 to it. And this is done, this reaction is performed by an enzyme called Rubisco. And that will come up and be important later. So Rubisco is the enzyme that catalyzes that reaction. And it's significant because really what we're doing, this is one of the most important chemical reactions on Earth for life, in that we're taking thin air, CO2, which is readily available in the atmosphere, and we're going to fix it into an organic form that can be used to build cells and organisms. And so we start, when we take 5 carbons and we add the 1 carbon from CO2, we get 6 carbons. So that's just basic carbon math. And almost immediately, in fact, this one is so short-lived, that 6-carbon intermediate, we're not even going to worry about the name of it. We're just going to say there's a 6-carbon intermediate. And that 6-carbon intermediate is very unstable and immediately breaks down into 2 molecules of 3-phosphoglycerate. And 3-phosphoglycerate, you may remember from our earlier study of glycolysis, is actually an intermediate in glycolysis during the energy producing stages. And so you should be a little bit familiar with this molecule. In the next step, we're going to take those 3-phosphoglycerates and we're going to make 1,3-bisphosphoglycerate. And we're going to make 2 molecules of that. So we had 2 molecules of 3-phosphoglycerate, and now we're going to have 2 more molecules of 1,3-bisphosphoglycerate. Now, in this process right here, you can ask yourself what happened. Well, we went from just a phosphoglycerate to a bisphosphoglycerate. So in doing so, we had to pick up a phosphate from somewhere. Where did that phosphate come from? Well, we took ATP and converted to ADP, and that's where the phosphate, the second phosphate came from, was from taking ATP. And of course, since we had 2 3-phosphoglycerates, we needed 2 ATPs to make our 2 1,3-bisphosphoglycerates. Where did this ATP come from? That was what was produced over in the light reactions that we talked about earlier. Now, in the next step, we're going to take our 1,3-bisphosphoglycerate and we're going to turn it into glyceraldehyde, which is also a glycolysis intermediate. Glyceraldehyde 3-phosphate. Okay. Now we've gone from a glycerate to a glyceraldehyde, and that's actually an oxidation step, and this is where we use our second reactant, which is going to be NADPH, and that's where this is used in this step, and we produce NADP+. We've got two molecules here, so we're going to actually use two of these. And we're going to produce two glyceraldehyde 3-phosphates. So this is our second product from the light reactions that we discussed earlier. And now we are going to use those in the Calvin cycle for this reaction. So now we've used some of our ATP, and we've used our NADPH as well. Now, in this next step, we're going to take one of these glyceraldehyde 3-phosphates, and we're going to transport it outside of the chloroplast into the cytosol. Right here, instead of writing glyceraldehyde 3-phosphate, I've just given a common abbreviation, and we just call it G3P, glyceraldehyde 3-phosphate. So we're going to take one of our glyceraldehyde 3-phosphates and move it that way. And now with the rest, I'm going to put 1x here for a second in quotations. Our next step is now we're going to take the other one and try and get back to our ribulose bisphosphate. Remember, because this is a cycle, it has to go over and over and over again. But we do face a bit of a problem in that up here, ribulose bisphosphate is a 5-carbon molecule, and our G3P over there only has 3 carbons. And so there's no real way to take a 3-carbon and turn it into a 5-carbon, at least not using normal chemistry. And so in order to solve this problem, what we had to do, the real problem came because we removed half of our product over here. The way then we make the Calvin cycle work so that we can balance it is we just start with more ribulose bisphosphate. So watch what happens. Instead of starting with one ribulose bisphosphate, I'm going to start with 3 ribulose bisphosphates. Now, remember, each ribulose bisphosphate needed a CO2, so let's start with 3 CO2s as well. We come over here, we're going to have 3 6-piece intermediates. And now, instead of having 2 3-phosphoglycerates, we're going to have 6. And we'll change all these numbers. We're going to have 6 of those, 6 of these, 6 of these. We'll go ahead and change up here the amount of ATP and NADPH now is also going to go up. So we're going to need 6 and 6 and 6 and 6. Now watch what happens. So we're going to still pull just one of them off. But now, instead of having just one remaining glyceraldehyde, we're going to have 5 remaining G3Ps. So we'll put here, there's our 5 remaining G3Ps. Now, when I do the math, remember G3P has 3 carbons on it, but I've got 5 of them, which means I've got a total of 15 carbons. Right? The 5 times 3. Now, up here, how many carbons did I start with? Well, I had a 5-carbon molecule, and I had 3 of them, so that's 3 times 5. So I had 15 carbons up here, too. So now the math works out. I've got 15 carbons down here in a G3P form, and I need to get to 15 carbons up here in a ribulose bisphosphate form. But at least we've got all the carbons there, the same number. So we start to have a series of complex chemical reactions, and we're not even going to go over any of them or what they are. We're just going to call this the ribulose regeneration stage. And we're not even going to talk about exactly what's happening in there, other than one feature. And that feature is this recombination of G3Ps to form ribulose bisphosphates does require us to have some ATP. So we do need ATP. We form ADP So it requires some additional phosphates. And those are going to come from ATPs. And the numbers are actually, it takes about 3 ATPs to make this conversion of 15 carbons into 15 carbons of ribulose bisphosphate. Okay? And so that is basically the Calvin cycle and how it works in its most reduced form, where we're only pulling off one G3P. Now, to actually produce a glucose molecule inside that plant cell, we would have to go through this whole thing twice so that we produce 2 G3Ps that could then be made into a 6-carbon glucose. Now, let's look at, for a moment, the stoichiometry of this process. And so on one side, we're going to look specifically at the ATP and the NADPH that are produced. So we've got the Calvin cycle right here, and it's going to require, what do we say? We need 6 NADPHs to go through that one time, and we need 9 ATPs. And we need 9 ATP molecules. And that doesn't seem like it's a problem, except for remember that the NADPH and the ATP, where did they come from? They were coming from the light reactions. So if we start to look at the light reactions then, we remember said that for every NADPH, we said for every 2 NADPHs, essentially, we produce 2 ATPs. Now you're saying, well, that's not nearly enough, but we can run this a couple of times. And so let's increase this to 6 and 6. So as we produce 6 NADPHs, we also produce 6 ATPs. In other words, they're just produced at a 1-to-1. So that's great. Right here, I needed 6 NADHs, and here I supplied them. Here I supplied 6 ATPs, but we do run into a problem in that we need too many ATPs in the Calvin cycle. I only produce 6 over here, but I require 9. Remember, we had those additional ATPs required in the regeneration stage, and that's going to mess up our 1-to-1 balance. And so as the Calvin cycle runs, because it requires more ATP, they're being produced in equal amounts, but more ATP is used than NADPH. And so what happens is that as the Calvin cycle runs, you start to get an accumulation of excess NADPHs, and they start to build. And pretty soon as you build up the NADPH over here, you run out of NADP+. And when you run out of NADP+, it has some effects on the light reactions. And so this becomes a real problem in that there's not enough ATP. The problem is, over time, you have an ATP deficiency and an NADPH buildup. So we've got too much NADPH, and we don't have enough ATP to keep the Calvin cycle running. Now, the solution to this then, if this is the problem, the solution is something called cyclic electron flow. So, to understand what cyclic electron flow is, let's go back over to our map of the light reactions. And what we drew up here in blue ink was something called linear or non-cyclic electron flow. So non-cyclic electron flow is in blue ink. But here's what happens when the Calvin cycle starts to create this imbalance of NADPH and ATP. Remember up here, NADP plus reductase required NADP plus in order to accept these electrons and create NADPH. The problem is, as the Calvin cycle runs, we run out of NADP plus. It's all in this form. And so this shuts down. And so we get a blockage right here on NADP reductase. The electrons are on the protein, and they want to be donated to NADP plus. There's no NADP plus, so they just sit here on that enzyme. Ferredoxin comes over. It's got a couple of electrons. It usually donates to this. It comes up to NADP plus reductase and says, OK, I've got your electrons. And the reductase says, I've already got electrons. I don't need any more. Take them somewhere else. And that backs the whole thing up like a great big traffic jam. Everyone's trying to move their electrons forward to the next carrier, but the next carrier is already occupied. And so it backs up the whole system, and the light reactions shut down. However, fortunately, ferredoxin right here, while it prefers to donate to NADP plus reductase, it does have another partner that it can go to. So the ferredoxin can actually come over here and donate its electrons as a secondary measure to plastoquinone. Now, if it donates to plastoquinone, same thing happens. We're going to go through here and donate the electrons to plastocyanin. Plastocyanin donates them to P700 plus. We go through the P700 cycle, and we get an electron, goes back to ferredoxin. We're having photons of light. We have to have these photons of light continually running, energizing these electrons on the P700 reaction center to keep this cycle going. But what you can see already is perhaps why this is then called cyclic electron flow. So we'll write that in black, cyclic electron flow. And what we're basically going to do is cut off the two ends. We're going to take photosystem 2 and the water splitting complex. We don't need them for cyclic electron flow. And we don't need NADP plus reductase. And we remove it from cyclic electron flow. But we do then just start cycling the electrons through this pathway that we've illustrated in black. Now, at first you might wonder, well, what's that do? How does this help the problem? What it's doing is it's running or powering the cytochrome BF complex. That's the key. The cytochrome BF complex continues to pump protons into the thylakoid space. So we'll draw a black arrow there. That's the key thing. And then these protons right here that get pumped across are going to move through ATP synthase back into the stroma, and we are going to continue to generate ATP. We don't get NADP plus, but we do get ATP. That ATP then comes over and fills up that deficiency. And as we build up our ATP levels, then the Calvin cycle will take the additional NADPH that we built up, and they're going to move it through and take care of our problem. And so that's how cyclic electron flow is able to overcome the stoichiometry problem of the Calvin cycle. And so the light reactions, if we come back over to the light reactions, the light reactions then will fluctuate between cyclic electron flow and non-cyclic electron flow based upon the availability of the NADPH as it passes through the Calvin cycle. And so that's how we're going to use cyclic electron flow to overcome our stoichiometry problem. So now the second problem that we said we were going to talk about is photorespiration. So let's talk about that for a second. So photorespiration is a playoff of two words. Photo coming from photosynthesis, because it's going to happen in the chloroplast where photosynthesis occurs. And then respiration, well, this reminds us of cell respiration that we talked about earlier in the mitochondria. And so remember, in cell respiration, we were using oxygen. That was the main input in cell respiration. And that's what's happening here. So in photorespiration, we don't use it, but the plant uses O2. Plant uses O2 instead of CO2. And that's in the Calvin cycle. And so what happens if we come over here and we look at the Calvin cycle, if we use, so let's say we don't use CO2, we use O2 in the Calvin cycle. So we take our 5-carbon ribulose bisphosphate and we add O2 to it. And it turns out that Rubisco, the enzyme that carries out this reaction, is a very ancient enzyme. And it really can't distinguish very well. Both of these are small gases, and it can't distinguish between CO2 and O2. And so it will take either one, even though it's supposed to take CO2. It will actually accept and add O2 to ribulose bisphosphate. But when you do that, you don't get a 6-carbon intermediate because you're not adding an additional carbon. You get a 5-carbon intermediate. And that 5-carbon intermediate, instead of producing 3-phosphoglycerate, produces 1 3-phosphoglycerate and 1 2-carbon molecule. And the 2 and the 3 equal 5. Now, the problem with that is as we go through then, we don't get 6. We get 3x, 3x, and 3x. This 2-carbon intermediate, we get a bunch of them, but it's totally useless. This has to go back out to the mitochondria to be used. It won't replenish or help us in the Calvin cycle. And so now we get down to this point. We were going to pull 1x off, but we don't have any more. So you're down to, if you pull off 1x, you get 2x left. And then we are back to that same problem of we now do not have enough carbons to regenerate our ribulose bisphosphates. We've lost carbons right here that had to be transported out. And so that represents a real problem. So when the Calvin cycle starts to take in oxygen instead of CO2, this whole process breaks down. The Calvin cycle completely shuts off because it can't regenerate ribulose bisphosphate anymore. Now, you might be asking yourself, so that's what happened. That's what photorespiration is. So the question that at least comes to my mind when I think about that is, why? Why would you use O2 instead of CO2? Why make that choice? Well, the plant is making a choice, not really to use O2 instead, but this is the problem that it faces. Photorespiration occurs when it's really hot outside. Now, when it's really hot, that means the sun is shining and hitting the plants. At first, you'd think, oh, that would be really good for photosynthesis because we have lots of light to drive the light reactions and make the whole photosynthesis process happen. However, when it is hot outside, it does drive photosynthesis, but it also drives a process that you know as dehydration, meaning simply that you get loss of water. So the plant has breathing holes. So we have stomata in the plant, which is equal to their breathing holes. And it's through those breathing holes that they intake CO2. So CO2 comes in. They get rid of O2 that they're creating. But H2O, or water, is also able to pass through those stomata when they're open. And so during the day, it's hot. If the stomata are open, we can exchange these gases. We can get rid of our excess O2. We can take in new CO2. The plant can essentially breathe. However, it also loses water vapor through those stomata. And if it loses too much water vapor, then the plant will dehydrate and die. So during the hottest part of the day, when it's really hot, the plant actually closes the stomata. And they close the stomata in order to save themselves from dehydration. So this prevents dehydration. The problem is that it also prevents gas exchange. CO2, O2 exchange. And so as the Calvin cycle and the light reactions start to happen, then the CO2 levels, they're being consumed, so CO2 levels go down, and the oxygen levels, remember in the light reactions, we produce oxygen by splitting water. So oxygen levels go up, CO2 levels go down inside the chloroplast. And that's what allows the rubisco, because now there's such this buildup of oxygen and all the CO2 has been decreased, that's what allows photorespiration. And so that's the photorespiration problem. It comes partially because of dehydration, loss of water. It comes partially because rubisco, the enzyme that carries out that fixing reaction, is not a very good enzyme for distinguishing between CO2 and O2. So, how do plants deal with the photorespiration problem? So, solutions or strategies. Strategies for photorespiration. So, the very first strategy we refer to as a C3 strategy for photorespiration. And the C3 strategy is to do nothing. So C3 plants, they don't really have a solution. They just stop photosynthesis during the hottest parts of the day. They close their stomata and shut down photosynthesis. And that's not a bad solution to it, but it does mean that photosynthesis in those plants is not occurring really at the best time for photosynthesis to occur. So when the sun is out and it's hot and it's shining down and you've got all this light that you can harvest, it shuts down because of photorespiration. And this is a majority, actually, of our crop plants. In fact, a majority of plants are C3 plants. So when you look at things like wheat, wheat is a C3 plant. Most crops are. Rice is a C3 plant. You can look at almost every kind of tree. There are C3 plants. And so most of the major, so things that you grow out in your garden, tomatoes, potatoes, carrots, all of those are C3 plants. They're shutting down photosynthesis at the best time of the day to do photosynthesis. Now, there's another group of plants that we call C4 plants. So C4 plants are going to use a different strategy to overcome photorespiration. And C4 plants are going to use an enzyme. So they use a different enzyme called PEP carboxylase. And PEP carboxylase can also fix CO2. It doesn't work in the Calvin cycle. So what they do is they fix CO2, but this enzyme is actually really good at recognizing even small amounts of CO2. And so what they do is they are going to harvest. So if you have a leaf cell, and right here you have the cells in the middle, and this is where you have the photosynthesis happening. And so PEP carboxylase exists in these other cells around the side that still have, remember in these photosynthetic cells, we've depleted all the CO2. But there's still some CO2 that had come in and was saturating these other cells up here. And this is where we're going to use PEP carboxylase, and we're going to take the CO2 out of these cells and transport it into these cells and then release it, again, right near the chloroplast of these mesophyll cells in the middle that are carrying out the photosynthesis. So we're going to fix CO2 in adjacent cells using PEP carboxylase and then transport and release that CO2 near the chloroplast in our photosynthetic cells. And so now we're going to produce a high concentration, and this allows the photosynthetic machinery to run for a little bit longer. You'll probably still eventually run out of CO2, but it allows it to get more photosynthetic activity out of the same sunlight because they're not shutting down, or at least they're delaying their shutdown. So C4 plants tend to be grasses that are associated with warm, dry climates. So some common C4 plants are, some common ones that are associated with warm, dry climates. Corn is actually a C4 plant. Sugarcane, fortunately, is a C4 plant. There's some other ones. Interestingly, papyrus is a C4 plant. And notice that these ones, for example, when you compare wheat and its growth cycle with corn and its growth cycle, so in the same season, you'll look out in the farmer's fields and the wheat will be growing this high and the corn will be growing this high. How does it do that? Well, corn is a C4. It's actually producing more sugar per sunlight or exposure to sunlight than the wheat can. Same thing with sugarcane. You grow these great big long stalks during the season. How can it do that? C4. And so that's the C4 strategy. We're going to use a different enzyme to fix it and then deliver CO2 to another site. There is a third strategy that we call the CAM strategy. CAM is going to be very similar to C4. This is used by succulent plants, things like cactus, actually pineapples do this. Okay, and there's some other plants that do this. And in the C4 strategy, again, we're going to fix carbon. And this one, the C4 plants are going to, at night, so their strategy is at night, they open their stomata. It's dark out, and hence it's probably cooler. And so at night, they open their stomata, and they'll breathe in a whole bunch of CO2, and they will fix that CO2 using an enzyme. They don't use PEP carboxylase, but it's similar to it. So they're going to fix CO2 and store it in their central vacuole, which has a lot of storage space. And so they're going to fix it as organic carbons inside the central vacuole, and then during the day, they are going to close their stomata and release CO2 from those organic compounds in the central vacuole. And that's the CO2 that they're going to use. And so this is just a second strategy. This is what we call a temporal strategy. They're opening at night and then fixing it and storing the CO2 as organic carbons, releasing it during the day when their stomata are closed. Up here, this isn't a night and day thing. This isn't temporal. This is a spatial separation, the C4 plants. Again, they're taking and they're gathering the CO2 that's in other cells around the photosynthetic cells and fixing it and then transporting in it. And this is all occurring during the day. And so those are some different strategies then that we have for dealing with the photorespiration problem. And this will conclude then. So if we come over here, a reminder, we've reviewed the light reactions. We've introduced the Calvin cycle and its stages. In fact, let me go back and do one thing that we didn't do earlier. We did do this a little bit. But we talked about one of the stages, the ribulose regeneration stage. But we didn't quite emphasize the other stages or even name them. And so the other stages right here, this first stage, this is called the fixing stage. It's only one reaction, the CO2, so this is called the CO2 fixing stage. It would form that part of the triangle. And then this second stage, which includes this 3-phosphoglycerate, the bisphosphoglycerate, the glyceraldehyde, that's referred to as the reduction stage of photosynthesis. As we create, it's really involved with the creation of glyceraldehyde phosphate, so that's the second stage. And then the third stage we did go over ribulose regeneration, and that would be this region right here. So those are the 3 stages. All right, so now we can come back over here and say that we have gone over the Calvin cycle and its different stages. We've talked about the stoichiometry of NADPH and ATP in both the Calvin cycle and the light reactions, and how we need the cyclic electron flow to be able to equilibrate the stoichiometry of those 2 product reactant pairs. And then finally, we talked about photorespiration and some of the different strategies that different types of plants use to overcome photorespiration. That's it for today. We'll see you next time.